discover exactly how our human brains came to be so highly connected, I've come to America to find out about the latest research into the human genome, the recipe for making a human. There are 3 billion letters in the human genome, stored in the 23 chromosomes that hold this recipe in every cell of our bodies. Each letter, A, G, C and T, represents one of the 4 bases, the chemical building blocks, which make up the long strands of DNA. And for geneticists like Frank Polow, these letters hold the clues that could unlock the secrets of the human brain.
So this is chromosome 1 written out.
Uh-huh.
And how much of chromosome 1 is it?
Just 1/50th.
And there's 1,000 pages here.
1,000 pages. 4,500 base pairs per page.
The whole genome would fill 670,000 sheets of paper. And at this rate, it would take me and Frank more than a week working 24 hours a day to lay out the entire human code.
It's amazing to think that our entire life, lies in this code.
And if we want to find out what makes the human code unique, we need to compare our own recipe with others. The breakthrough that lets us do this is that, as well as the human genome, we now have sequenced the genomes of many other animals. But finding the crucial sections of code that make us human is a monster puzzle.
One of the important steps in this process to try to identify what in our genome, in the human genome, could underlie what makes us human is to try to find differences at the base pair level in the coding sequences between us, basically our genome, and the genome of our closest living relative, at least, the chimpanzee.
Frank has homed in on one particular change that is specific to humans, which he believes could be fundamental. It involves a gene called SRGAP2 that is found in all animals and mainly affects the developing brain. But in humans, and only in humans, this gene is duplicated 4 times.
This gene starts at page 814, and the very beginning of the sequence is this sequence, CACAGGAA. And so the gene starts here.
I can't believe you can recognise that.
The gene is about 125,000 base pair long. So it goes from page 814 to 840.
So that is a single gene.
That's a single gene, exactly. And the sequence basically ends right here, TGC, TGC, GT.
So this is a gene that we have in common with the other apes, but we've got 3 more copies of it.
Correct. We've got 3 more copies of this gene. They would be in volume 30. Remember, this is only one volume for chromosome 1. The full sequence of chromosome 1 would take about 50 volumes, right?
Right.
So the copies would be in volume 30, 31, 32.
What Frank discovered was that the human duplication of SRGAP2 has a dramatic effect on the connectivity of neurons. By splicing the human duplicate into mouse DNA, he showed that the mouse neurons increased their ability to form connections.
So this is a normal mouse brain, and that's what happens if you put that
SR-gapped.
duplicated bit
Exactly.
of SR-gapped2.
You form many, many more spines, basically, and we have other evidence to show that those neurons are actually hyper-connected. You increase by about 40% the total number of connections made onto these neurons. Why is it so exciting? Basically, humans stand apart completely. Human neurons have about 40 to 50% increase in the total number of connections made onto those neurons, which we know is a feature that sort of distinguishes human neurons, basically.
For me, this is when genetics gets really exciting, because we've got an actual observable difference in the brains of chimpanzees versus the brains of humans. And now we've got something in the genome which could explain that actual physical difference in our brains.
Exactly.
Every nuance of human behaviour somehow springs from this massive branching network of hyper-connected neurons in our huge brains. It's what makes the human brain so brilliant, this complex wiring diagram of connections that holds our memories, our emotions, our ability to row a boat or to draw.